So without wasting much more of the time, I'm just starting off with today's class. And like previous day, we have discussed a few PYQs in case of what we studied the last day. Today also, we are just going to go through uh, some of the details of the GATE 2023, the exact questions from which the exact topics from which the questions are generally being asked in case of zoology portion of the GATE Excel 2023. तो हम लोग आज भी देखेंगे कि हम लोग क्या पढ़ने वाले हैं, तो तुम लोग ध्यान से देखना. रिकॉर्डिंग्स अवेलेबल रहेंगे यूट्यूब चैनल पर, और जो लोग अभी लाइव क्लासेस कर रहे हैं उनके लिए भी सुविधा होगा. So at the beginning we would be talking about antibodies. Now antibodies, as you all know, these are certain chemical molecules in the form of proteins that are produced in our body in response to something which is known as the antigens. Now antigens may be of several types, right? Now you may have a type of antigen which may be proteinaceous in nature. You may be having a type of antigen which can be carbohydrate in nature. So it varies. Okay, fats can act as antigens. Any foreign particle, it can be foreign or it can be self. Okay, so you should remember that all antigens are exactly not foreign, but there are several types of antigens which are also self antigens, which are denoted as self antigens. So now these self antigens are of very much importance because of the fact that these antigens play massive roles in case of eliciting the immune system. Okay, that means activating the immune system. Now these antigens, whenever they activate the immune system, there are several mechanisms which the immune system is generally activated. Now you can be having the first line of defense, then you can be having the second line of defense. Now among these first line, second line of defenses, there are several types of strategies which are involved in this case. Like you might be having several types of cells which are involved, several collection of cells which might also get involved. But yes, on a whole, it is basically a final type of response which is generated by these proteinaceous molecules in our system that are inbuilt in nature, and these are known as these antibodies. Okay, so these are the antibodies in general. So these antibodies are designed by our own body. That means our body produces these antibodies. These antibodies are, I mentioned that these antibodies are nothing but proteinaceous in nature, and the exact processes via which the protein packaging takes place, that is the post-transcriptional modification takes place, is exactly similar to that of the antibody processing also. So now in this case, the antibodies are directed against a extracellular antigen molecule. Okay, so antigen molecule which is coming outside from our cell is being targeted by these antibodies, and these antibodies are said to act as a final line of defense against antibodies. These antibodies have against antigens. Now these antibodies have minimal roles via playing in the innate immune system, but they have the maximum roles when it comes about the acquired immune system. Okay, so these antibodies, among these antibodies, the most important ones are the IgG, the IgA, the IgM, the IgE, and the IgD. Now IgG has the highest opsonization and neutralization activity. IgM is the first antibody that transiently increases upon antigen invasion. So I have just jotted down few important points about this IgM, IgG antibodies. Now IgA is expressed in the mucosal tissues. IgE is involved in allergy. IgD functions as an antigen receptor on activated B cells. So please keep an eye on the functions of the GA, MED antibodies. G has the highest opsonization and neutralization activities. A has expressions in the mucosal tissues, that means the tissues which are involved in the collection of the cells which secretes mucus. Okay, the patchy and sticky type of liquid which has different types of functions to play at different organs of our body. Like they play an important role in the immune system by playing a role in the primary barrier of our immune system. You all know about the mucosal barriers that are involved in the primary defense against different types of pathogens which may enter our body. Again, this mucosa is also lined in the gut area of our body. Okay, so it may be the alimentary canal, it may be the intestines. You might be finding these mucosal tissues all round of our body, and they play a vital role in the immune system. Okay, again, M antibody, that is the IgM antibody, is the first antibody transiently increased upon antigen invasion. Whenever there is an antigen, it might be any kind of antigen which might be entering our body. The first type of antibody that is activated in our body is the IgM antibody, and this IgM is the largest antibody that is present in our system. Now the next one is the E antibody, and you know that the IgE antibody is generally involved in the immune response functions. Okay, and the type 1 hypersensitivity reactions is cornered around this IgE antibody only. Now the IgD antibody functions as an antigen receptor that is activated on the B cells. So antigen receptors, I have already mentioned that antigen receptors that are expressed on the B cells, that is especially the activated B cell, they are known as the B cell receptors, and these B cell receptors are generally in the form of this IgD antibody. Okay, so these are the basic functions and the basic acting organs or the basic acting cells of this GAMED Ig antibodies. Ig means immunoglobulin, and immunoglobulin is on a whole a large set of a superfamily of these antibody like proteins, not antibodies but antibody like proteins, immunoglobulins. There might be many kind of many kind of proteins that may be involved in this Ig superfamily or this immunoglobulin superfamily. Now IgG, IgE and IgD remain generally as monomer antigens. So you will look at the structure, we will look into the structure of these three or these 5 antibodies in details in the generalized section of the next slide, but you have to remember that IgG, E and D act as monomer, remain as monomeric antigens. The IgA remains as a dimer or a trimer. Okay, IgA remains as a dimer or trimer. IgM is a pentameric structure. Now IgA comprises of 2 classes, the IgA1 and the IgA2, while IgG has 4 subclasses, IgG1, IgG2, IgG3, IgG4. Now IgG1 is the most important of these 4 subclasses, and it plays the most of the roles that is being played by this IgG superfamily of the antibody. So IgG has 4 subclasses and IgA has 2 subclasses. So this is a bit of details about the antibodies, from where generally people used to ask the questions. Now this is basically the structure of the antibody. Now you can look over here that the antibody has 2 types of parts in general. This is a basic structure of an antibody. Okay, so and this is, you can say that this is a structure of a IgG antibody, and the IgG is the basic type of antibody that is present in our system. It has 2 parts in general. You can look over here, that is the Fab area and the Fc area. The Fab area is the fragment of antigen binding and Fc is the fragment of the constant interaction region. So fragment of antigen binding provides the binding ability of the antibody to the antigen, and the fragment of constancy is where there is the constant sites which are present, and in the fragment of antigen binding the variable sites are generally present. Why? Because antigens might be having different types of membranous, or you might be saying different types of topography means उसके सरफेस पे जो सारे मॉलिक्यूल्स एक्सप्रेस्ड होते हैं वो सारे डिफरेंट हो सकते हैं डिफरेंट टाइप्स ऑफ एंटीजेंस के लिए, लेकिन ये कभी नहीं होगा कि एंटीबॉडीज जो एंटीजेंस होते हैं वो डायरेक्टली जाके एफसी रीजन से इंटरेक्ट करें। नहीं, एफसी रीजन से कौन इंटरेक्ट कर सकता है? एफसी रीजन से इंटरेक्ट कर सकता है कोई भी टाइप का सेल, यानी ऐसा हो सकता है मास्ट सेल के साथ एफसी रीजन ऑफ द आईजीई एंटीबॉडी इंटरेक्ट करता है, ठीक है। और दूसरे तरीके से बी सेल रिसेप्टर्स जो है, बी सेल के बाहर जो सारे रिसेप्टर्स है, हमने बोला था कि यहाँ पे लिखा हुआ है आईजीडी फंक्शंस एज एन एंटीजन रिसेप्टर ऑन द एक्टिवेटेड बी सेल, दैट मींस दे एक्ट एज एंटीजन बी सेल रिसेप्टर्स। ओके, सो ये बी सेल रिसेप्टर्स जो है, इन बी सेल रिसेप्टर्स ये आईजीडी एंटीबॉडीज बी सेल रिसेप्टर्स के थ्रू एक्ट करता है, बी सेल रिसेप्टर्स के जैसे एक्ट करता है, और ये बी सेल रिसेप्टर्स के जो एफसी डोमेन होता है, और ये एफसी डोमेन जनरली इंटरेक्ट करता है बी सेल के साथ। So there might be variable things in terms of the antigen binding, but yes, there is no variability in terms of the constant region. So variable regions can be found in two types of chains which are found in this antibody. Antibody light chain, this is the antibody light chain having the lighter molecular weight, and the antibody heavy chain which is having the greater molecular weight, that is more molecular weight, that is more heavier than that of the light chain. Now this light chain comprises of the variable region and the constant region, and this heavy chain comprises of the variable region and these three constant regions. So there are three constant regions that are present in the heavy chain, that is the CH1, CH2 and CH3. In case of light chain, it is only one, that is Cl. And in case of the variable region of the heavy chain, there is the VH region, and in case of the light chain, there is the VL region. The variable region comprises of the CDRs, that is the complementarity determining regions. Now these CDRs are generally the areas at which the paratopes are generally present. Now what are the paratopes? Paratopes are several sites or the several interacting areas via which, not areas actually, they are the different molecules that might be present in the form of amino acids or in the form of any other thing that might be interacting with that of the antigenic epitopes. Okay, the epitopes are certain molecules or certain structural patterns that are present on the, in case of the antigenic molecule. Now the antigenic molecules epitope interacts with the paratopes, and these paratopes are clustered inside the CDRs of the variable region in the FAP region of the antibody. So this is on a whole what is the structure of the antibodies. Now these antibodies are generally stabilized using different types of disulfide bonds. Now these disulfide bonds are linked to each other. Disulfide bonds are generally linked to each other using different types of interactions. Now these interactions can be heavier interactions, generally they are, because you know that the disulfide bonds are covalent in nature. Again, these disulfide bonds can also be broken using different types of enzymatic cleavages. It can be papain cleavage sites that might be present at the hinge region of the heavy chain, that is here, over here, see there is a hinge region that is present, and this hinge region can be cleaved using papain. Papain cleaves it into Fab and Fc. Okay, Fab and Fc can be cleaved by papain. There are pepsin cleavage sites that are also present over here, and here is the structure of the antibody on a whole. So I hope that you have understood this thing. Now let's move on to the next thing. So what I was talking about was about this structure, that is the IgM pentameric structure, the IgG monomeric structure, the IgA dimeric structure, IgE monomeric structure, and IgD monomeric structure. Now the IgM pentameric structure has heavy chains in the form of mu. IgG has a heavy chains in the form of gamma. IgA has in the form of alpha. IgE has the form in the form of epsilon, and IgD has in the form of delta. Now number of antigen binding sites in case of IgM pentamer is 10, but you have to remember that there are the total number of CDRs or the complementary trait determining regions in case of IgM is 60, around about 60. Okay, whatever is the amount of total number of total number of antigen binding sites that are present, you multiply those with 6 to get the amount of CDRs. So if you are asked how many number of IgM CDRs are present in a single IgM molecule, what is the minimum number of, you have to say it is, the minimum number is around 60. The options would be given in the form of 30, 40, 10. 10 would be there in an option, but the 10 is not correct actually, it's 60, 6. In case of IgG also there are 6. So basically there are 6 CDRs that are present in a IG or the immunoglobulin monomer. So there would be 12 CDRs. Again in case of the IgA there would be 24. IgE again 12, IgD again 12. Okay, in this way basically the number of antigen binding sites, you know that there are generally 2 antigen binding sites in a monomer of an antibody, right? I see in case of IgG there are 2, IgE there are 2, IgD there are 2, but the CDRs would be in the form of 6 into the number of antigen binding sites in case of different types of antibodies. So molecular weights are in the form of this. See, it has the highest molecular weight, and it is quite common that it would be having the highest molecular weight. IgE has the least molecular weight, IgE has more, and IgD has a bit more again, and IgA has a double molecular weight type. Okay, in this way the immunoglobulins are generally classified. So cross placental barrier is the IgG monomeric antibody because of its smallest size. Complement fixation is being shown by IgM and IgG antibodies. FC region binds to phagocytes in case of IgG monomeric antibody, IgG mast cells and basophils, and in case of IgD, I have forgotten to mention, it will be the B cell plasma membrane. Okay, B cell plasma membrane. I would be adding this one. Please remember this, B cell plasma membrane. Percentage of antibody in the serum, highest percentage is present in case of the IgG monomer. First activation is the IgM. Okay, first activation is IgM, but more number is present in case of IgG. Again, placental crossing is shown by IgG. Then complement fixation is shown by this IgG and IgM. FC region of IgG binds to phagocyte. FC region of IgG binds to the mast cells of the basophils. In case of the FC region of the IgD binds to the B cell plasma membrane. Function of IgM is main antibody of the primary responses best at fixing complement. The monomeric form of IgM serves as the B cell receptor. So the monomeric form of IgM is basically the IgD that acts as a B cell receptor. Please remember this point. The monomeric form of IgM is the IgD. Now you can also check this out over here. See the molecular weight of the IgD. 18000, multiply it with 5 and you will get 900000. Look over here, it's 900000. 5 take 5 IgD monomers, multiply it with 5, you will get this one. So basically IgM pentamers, if they are digested with certain amount of this oxidizing agents, which the reducing agents, and you will get something, a strong reducing agents that would break the disulfide bonds, the disulfide interlinkages, and you will find out that these IgM pentamers are present in the form of IgD monomers. So the next point is about the IgG monomer. Main blood antibody of the secondary responses, neutralizes toxins and helps in opsonization. IgA is secreted into the mucus, saliva and colostrum. So IgA antibody is present in case of colostrum, milk. That is the yellowish color of the milk that is present at the beginning. Okay, so that's all. Then the next thing is present in case of IgE monomer, antibody of the allergy and anti-parasitic activity. B cell receptor is present in case of the IgD monomer acts as basically the B cell receptor, and the IgE monomer acts as a allergen or the allergic component antibody that is present in the surfaces of the mast cells of the basophil. Next is basically about the class 1 and class 2 of the MHCs. Now you first have to know what is MHC. MHC is nothing but the major histocompatibility factor. Now the major histocompatibility factor has different types of chromosomal components that from which it might be arising. Now in case of humans there are different types of chromosomes that are involved. In case of mice there are different types of, but in case of humans they are known as the HLA factors. Now, or it is also known as the human leukocytic antigen factor. Now what are these MHC class 1 and class 2? Now whenever there is an antigen that is present in the environment, different types of cells are present that are known as the APCs. What are APCs? APCs are the antigen presenting cells. So they are generally classified into two types. So there are two types of APCs that might be present, that is the professional APCs and the non-professional APCs. Professional APCs are something like you are generally engaged in a profession. That means that you might be doing a PhD, you might be a teacher of a college or a school, you might be something else, you might be engaged in a different kind of profession, you might be a laborer, you might be a worker who is working in the industries, you might be something else or a IT professional or something. So you are engaged in a profession. But some people are generally not engaged in a one-directed profession, that they are the non-professional people. Okay, so they might be doing some kind of freelance works, they might be doing a job of a technician, they might be doing a job of, they again might be doing a job in a cyber cafe. So in this way, some people do like to have jobs which are generally professional jobs, and some people do not like to have jobs that are the professional jobs. They are classified under non-professional. So professional means they are engaged in something in which they are mostly they are likely to do that. Okay, so हम लोग ये कह सकते हैं कि एक मजदूर को मजदूर की काम ही पसंद है, एक पसंद नहीं है, वो काम ठीक से कर सकता है। अगर किसी पीएचडी स्कॉलर को अगर मजदूर का काम दे दो या फिर मजदूर को पीएचडी स्कॉलर का काम दे दो, कोई भी ठीक से नहीं कर पाएगा। तो जिसका जो काम है वो ठीक से कर सकता है। तो इस प्रोसेस को हम लोग प्रोफेशनलिज्म के नाम से जानते हैं। और ये प्रोफेशनलिज्म या फिर प्रोफेशन वाइज जो एंटीजेंस या फिर जो एपीसीज काम करते हैं, नॉट एंटीजेंस, जो एपीसीज काम करते हैं उनको हम लोग प्रोफेशनल एपीसीज के नाम से जानते हैं। और ये प्रोफेशनल एपीसीज जो है ये जनरली शो दिस प्रॉपर्टीज जनरली फाउंड बाय 2 or 3 types of antigen presenting cells or the immune cells. That is, one is the macrophage, one is the neutrophils, another one is the dendritic cells. There may be several other types, but these 2 or 3 are very much important. Now, non-professional antigen presenting cells can be any cells which comprises of the nucleus. Okay, any cells that comprises of the nucleus can be antigen presenting cells, which are of the non-professional type. So these antigen presenting cells, whenever they, especially this professional or non-professional antigen presenting cells, whenever they get some kind of pathogen, they process those pathogens. That means they take in those antigen fragments, they process those antigen fragments, and they display those antigen fragments with the help of a kind of a complex that is known as the major histocompatibility complex to a certain kind of a cell, and that cell is generally in the form of the T cells. Okay, T cell के फॉर्म से ये सारे एंटीजन प्रेजेंटिंग सेल्स डिस्प्ले करते हैं उन एंटीजेनिक फ्रैगमेंट्स को। और ये एंटीजेनिक फ्रैगमेंट जब टी सेल के पास डिस्प्ले होता है, टी सेल वहाँ से एक रिसेप्टर निकलता है जिसका नाम होता है टी सेल रिसेप्टर, एंड देयर आर सेवरल co-stimulatory molecules or co-inhibitory molecules that are involved in the signaling pathway of the interaction between the professional or non-professional antigen presenting cell and the T cell. Now this interaction plays a very important role in paving the way for the further downstream immune responses. Now these MHCs or the major histocompatibility factors are the ones which holds the antigenic fragments to the T cell receptors. They are divided into two classes, that is the class 1 and the class 2. Now the class 1 is basically encoded comprised of an MHC encoded chain and a beta 2 microglobulin chain, and the class 2 comprises of a alpha and beta chain. That means a pair of alpha and a pair of beta chains are present in class 2. But in case of class 1, what is present are 3 alpha chains, that is alpha 1, alpha 2, alpha 3 and beta microglobulin chain. So basically this MHC class 1 is present on most type of cells. That is, it is present on the non-professional antigen presenting cells as well as the professional antigen presenting cells. But in case of MHC class 2, it is only present in the antigen presenting cells. That means what type of antigen presenting cells? They are present on the professional antigen presenting cells. You have to remember this. Bind endogenous antigens synthesized in a cell. Bind exogenous antigens. See, antigen presenting cell, that is the professional antigen presenting cell, only binds to exogenous antigens, and they might bind to endogenous antigens in a cell. Okay, exogenous antigens means जो भी पैरासाइट्स या फिर एंटीजेंस बाहर से आ रहे हैं, बॉडी के बाहर से, सेल के बाहर से आ रहे हैं। लेकिन एंडोजिनस एंटीजन का मतलब है सेल के अंदर सिंथेसाइज हो रहा है। तो अगर तुम लोगों को पूछा जाए कि एक वायरस अगर बाहर से आया और वो जीनोम के साथ इंटीग्रेट कर गया, जीनोम के साथ इंटीग्रेट कर जाने के बाद जो जीन प्रोडक्ट्स वो निकलता है, वो जीन प्रोडक्ट्स किस फॉर्म में आएंगे? वो होगा एंडोजिनस एंटीजेंस के फॉर्म में, यानी इट इज यूजिंग द cellular mechanism of our own body for the synthesis of its own gene products. So this might be in the form of the endogenous antigens. But exogenous antigens मतलब बाहर से कोई पैरासाइट डायरेक्टली एंटर कर, लाइक वायरस माइट बी एंटरिंग आवर बॉडी और उसके बाहर जो कैप्सिड लेयर है वो टूट जाता है, देन that broken capsid layer is taken up by the macrophages or the dendritic cells or the B cells. It might be taken up by anything, and when it receives, it breaks it into fragments and displays via the MHC class 2. So MHC class 1 and class 2. Now this MHC class 1 antigen presents antigen to cytotoxic T cell lymphocytes. Now MHC class 2 antigens present it to the helper T cell lymphocytes. Helper T cell lymphocytes have a CD4 adhesion molecule that is present, and the cytotoxic T lymphocytes, that is the CTLs, are having a CD8 marker molecule. That is CD8 is present on CTLs and CD4 is present on T cells, that is T helper cells. Presence of foreign or overabundant antigens targets cell for destruction. Now MHC class 1 जो सारे सेल्स बियर करते हैं, वो अगर MHC क्लास 1 के थ्रू कोई भी एंटीजन का डिस्प्ले करता है, तो वो जो टी सेल होगा वो सोचेगा, अच्छा ये वाला CTL, ये वाला जो है ये जनरली मुझे डिस्प्ले कर रहा है एक ऐसा एंटीजेनिक मॉलिक्यूल जो एंडोजिनसली सिंथेसाइज हुआ है. वायरस इन्फेक्टेड हो सकता है, कैंसर सेल्स हो सकता है, तो CTL का काम होगा MHC क्लास 1 जो भी सेल डिस्प्ले कर रहा है उसको मार दो, CTL का काम. या फिर वो टी हेल्पर सेल को एक्टिवेट कर सकता है, टी हेल्पर सेल आके यहाँ पे कोई रिपेयर पाथवे को भी एक्टिवेट कर सकता है. लेकिन जनरलाइज पैटर्न से ये सेल डिस्ट्रॉयड किया जाता है बाय द हेल्प ऑफ द साइटोटॉक्सिक टी लिम्फोसाइट फैक्टर्स इन द फॉर्म ऑफ परफोरिन ग्रांजाइन. यू नो अबाउट दिस एंजाइम. ना MHC क्लास 2 में जो होता है, प्रेजेंस ऑफ फॉरेन एंटीजेंस इंड्यूसेस एंटीबॉडी फॉर्मेशन. सो टी हेल्पर सेल जो है बी सेल को एक्टिवेट करता है. बी सेल किससे एक्टिवेट होता है? ये सारे एक्टिवेशंस जनरली होते हैं इन द फॉर्म ऑफ आइदर इन द फॉर्म ऑफ रिसेप्टर्स और इन द फॉर्म ऑफ सीक्रेटेड सॉल्युबल फैक्टर्स इन द फॉर्म ऑफ साइटोकाइंस, केमोकाइंस जैसे मॉलिक्यूल्स हैं. तो बी सेल्स को एक्टिवेट कर देता है. देन बी सेल्स एंटीबॉडी सिंथेसिस करता है या फिर कॉम्प्लीमेंट प्रोडक्शन करता है. उसके माध्यम से ये जो है ऑप्सोनाइजेशन, कॉम्प्लीमेंट फिक्सेशन के माध्यम से जो बाहर से आने वाले एंटीजेंस है ये सारे न्यूट्रलाइज कर दिए जाते हैं. तो इन दिस वे MHC class 1 and MHC class 2 was very important in terms of GATE or any other exams. MHC class 1 and MHC class 2. Please remember these things what I have told in the class. So next thing is about red blood cells. That means red blood cells के बाहर कुछ लेयर्स पाए जाते हैं, या फिर रेड ब्लड सेल्स के ग्रुपिंग में नॉट एग्जैक्टली लेयर्स, तुम नहीं बोल सकते लेयर्स पाए जाते हैं, ऐसा कुछ नहीं है. बट येस दे डिस्प्ले सम काइंड्स ऑफ प्रोटीन्स ऑन इट्स आउटर सरफेस, और एक्सटर्नल सरफेस में बहुत सारे प्रोटीन्स होते हैं प्लाज्मा मेम्ब्रेन के बाहर, या फिर अंदर भी बहुत सारे प्रोटीन्स. येस दे आर बीइंग फाउंड, बट येस दे डू नॉट कंप्राइज ऑफ अ न्यूक्लियस. यू हैव टू रिमेंबर दिस थिंग इन माइंड. द रेड ब्लड सेल्स के पास न्यूक्लियस नहीं होता है. एक एनिमल है जिसके रेड ब्लड सेल्स में न्यूक्लियस होता है, उसका नाम है कैमल. कैमल के पास रेड ब्लड सेल्स के अंदर न्यूक्लियस होता है. बट इन केस ऑफ ह्यूमंस देयर इज नो न्यूक्लियस इन केस ऑफ रेड ब्लड सेल. So how is the grouping in case of the red blood cells done? We talk about the groups like in the form of AB and O. Okay, so how is this grouping done? Now this grouping is done by relating the antigens that are present in the red blood cell. If A antigen is present, look over here, if A antigen is present, then the red blood cell type is grouped as group A, that is A. If B antigen is present, red blood cell is termed as B. If both A and B antigens are present, it is termed as group AB. If no antigen is present, it is termed as group 0 or O. It is basically 0, but we terminalize it as O. Okay. So यहाँ पे देखो बाहर में जो है यहाँ पे A एंटीजन डिस्प्ले कर रहा है ये रेड ब्लड सेल, तो इसको हम लोग ग्रुप A या फिर A के नाम से जानेंगे. अगर बाहर B एंटीजन डिस्प्ले करे तो उसको B एंटीजन के नाम से जानेंगे. अगर बाहर दोनों एंटीजन देखो एक साथ है यहाँ पे, तो इसको हम लोग AB एंटीजन दोनों एक साथ अगर है तो इसको हम लोग AB ग्रुप से, और यहाँ पे अगर कोई भी एंटीजन नहीं है बाहर तो उसको हम लोग ग्रुप O के नाम से या फिर ग्रुप 0 के नाम से जानेंगे. तो अगर बाहर ग्रुप ए के बाहर अगर ए एंटीजन डिस्प्ले होगा तो प्लाज्मा यानी रेड ब्लड सेल्स के पास यानी कि ब्लड के पास जो सिस्टम होता है वहां पे देखा जाता है कि टू टाइप्स ऑफ थिंग्स आर देयर, दैट इज द सेल्स एंड द प्लाज्मा. ओके, सो सेल्स के अलावा ब्लड में जो चीज़ होता है उसको हम लोग प्लाज्मा के नाम से जानते हैं। प्लाज्मा ही है जहाँ पे एंटीबॉडीज पाए जाते हैं। सो प्लीज रिमेंबर, प्लाज्मा इज द ओनली प्लेस वेयर द एंटीबॉडीज आर जनरेटेड। होल ब्लड में तो सेल्स भी है, तो होल ब्लड, हम लोग ब्लड को अगर कोई भी पेशेंट को एंटीबॉडीज़ हमको देना है किसी डिजीज़ के वजह से, अगर कोई एंटीबॉडीज़ हमारे शरीर में जनरेट हुआ है, हम लोग कोविड के वक्त में देखे थे कि कॉन्वल्सेंट प्लाज्मा थेरेपी एक बहुत प्रचलित टेक्निक था उस वक्त, ठीक है। तो वहाँ पे जो होता था कि प्लाज्मा को कलेक्ट किया जाता था, ठीक है, वेट बाय ग्रेविटी मेथड से प्लाज्मा को पहले कलेक्ट किया जाता था और प्लाज्मा को फिर दूसरे पेशेंट पे डोनेट किया जाता था। तो प्लाज्मा इज वेरी मच इंपॉर्टेंट इन टर्म्स ऑफ द of being found with the abundance of the antibodies, okay। सो एंटीबॉडीज का जो अबंडेंस है वो प्लाज्मा में ही देखा जाता है। सो ग्रुप ए के यहाँ पे प्लाज्मा में देखा जाता है कि एंटी बी एंटीबॉडी प्रेजेंट रहता है, ऑपोजिट ऑफ व्हाट इज प्रेजेंट, ओके। एंटीजन जो एंटीजन रहेगा यहाँ पे, ठीक है, जो भी एंटीजन रहेगा, यानी कि इट माइट बी इन द फॉर्म ऑफ एंटीजन ए। If antigen A is present, then the antibody that would be present is anti-B antibody. If B antigen is present, then anti-A antibody is present in case of the plasma in the antibodies. If both A and B antigens are present, no antibodies in plasma will be present. If no antigens are present, then anti-A and anti-B antibodies are present. So please remember this thing. This is very much easy, and from this concept questions have been asked approximately 4 or 5 gates ago. But yes, the next in the very next one, that is 2023, a question might just pop up from the past 6 years, and from this year a question may be coming from this area. So next thing हम लोग जो डिस्कस करने वाले हैं वो है ऑटो इम्यून डिसऑर्डर। Okay, so there are several types of diseases that might arise due to the malfunctioning of our immune system, where our immune system targets our own cells. Now these type of disorders are generally known as this autoimmune disorder. Now person may be having more than one autoimmune disorder at a time, okay। एक ही समय में एक पर्सन के पास 2 autoimmune disorders हो सकते हैं। कुछ autoimmune disorders का नाम है एडिसन्स डिजीज, ग्रेव्स डिजीज, मल्टीपल स्क्लेरोसिस, मायस्थीनिया ग्रेविस, pernicious anemia, rheumatoid arthritis, and then systemic lupus erythematosus. Addison's disease can develop if the immune system attacks the adrenal glands. अगर adrenal glands, especially adrenal cortex को directly attack करता है immune system, तो ये हो सकता है कि adrenal cortex के वहाँ पे जो cells है वो cells destroyed हो जाए। अगर cells adrenal cortex के destroyed होते हैं तो तुम लोगों को पता है कि adrenal cortex is generally responsible for the synthesis of steroid hormones such as cortisol, aldosterone, and these synthesis may be disrupted, and there might be several types of diseases which might be arising in the form of a collective bracketed disease. It is known as the Addison's disease. Next thing is known as the Graves disease. ग्रेव्स डिजीज एक बहुत ही इंपॉर्टेंट फिनोमेना है और यहाँ पे देखा जाता है कि थायराइड स्टिमुलेटिंग हार्मोन जो है, थायराइड स्टिमुलेटिंग हार्मोन के लुक अलाइक एक एंटीबॉडी प्रोड्यूस हो जाता है जिसका नाम होता है थायराइड स्टिमुलेटिंग इम्यूनोग्लोबुलिन, और इसके थ्रू जो होता है कि हाइपोथायराइडिज्म जनरली देखा जाता है। टीएसएच का काम है थायराइड ग्लैंड का साइज को बढ़ाना, और थायराइड ग्लैंड में से जो हार्मोन्स निकलते हैं यानी T3 T4 ऐसे हार्मोन्स का जो है सिंथेसिस रेट्स बढ़ा देना। तो टीएसएच का ही लुक अलाइक हार्मोन इम्यूनोग्लोबुलिन या फिर एंटीबॉडी पाया जाता है, दैट इज द टीएसआई। और ये टीएसआई प्रोड्यूस होता है अगर हमारे शरीर में कोई डिजीज हुआ है, और डिजीज को काउंटर करने के लिए एंटीबॉडीज जनरली प्रोडक्शन होता है, और वो एंटीबॉडीज अचानक से टीएसएच हार्मोन के अलाइक हो गया है, लुक अलाइक हो गया है, ठीक है। सो ये हो सकता है कि टीएसआई एंटीबॉडी कॉज द थायराइड ग्लैंड टू प्रोड्यूस एक्सेस थायराइड हार्मोन, दैट hypothyroidism, okay। this is Graves' disease। मल्टीपल स्क्लेरोसिस जो होता है वो मायलिन शीथ के डैमेज की मदद से मल्टीपल स्क्लेरोसिस होता है। दिस शीथ इज अ प्रोटेक्टिव कवरिंग दैट सराउंड्स द नर्व सेल, यू ऑल नो दैट। व्हेन दिस नर्व कवरिंग इज डैमेज, nerve covering is damaged, nerve signals generally slow down, okay, they might also stop। The nerve damage is caused due to inflammation, and inflammation, as you all know, that it occurs when there is an attack on some kind of a cell by the immune system. जब भी immune complexes attack करता है तभी एक एरिया में immune system के द्वारा inflammation टेक प्लेस करते हैं. इन्फ्लामेशन मतलब कलेक्शन, इन्फ्लामेटरी एरिया में क्या होता है, कलेक्शन ऑफ बहुत सारे इम्यून सेल्स, इम्यून कॉम्प्लेक्सेस हो जाता है. एंटीबॉडीज हो सकता है, न्यूट्रोफिल्स, मास्ट सेल्स, मैक्रोफेजेस, सब एक जगह पे आके एक लंप एरिया बना देता है और उस लंप एरिया में रेडनेस हो जाता है, डेवलप होता है. लेकिन वो इन्फ्लामेशन जनरली कुछ ही दिनों में हील कर जाता है अगर जो कॉज ऑफ इन्फ्लामेशन है वो वहाँ से रिमूव कर दिया जाता है. तो ये इन्फ्लामेशन जो है ये जनरली टेक प्लेस करता है अगर हमारे बॉडी सेल्स जनरली किसी भी बॉडी का इम्यून सेल्स जो है किसी भी सेल्स को टारगेट करता है. तो इन केस ऑफ मल्टीपल स्क्लेरोसिस it is the myelin sheath which is under the attack of the immune cell. Okay, this can occur in any area of the nervous system, that is the brain, optic nerve and the spinal cord. Then there comes the myasthenia gravis. Myasthenia gravis जो है एक न्यूरोमस्कुलर डिसऑर्डर है. Now muscles, nerves दोनों ही इन्वॉल्व होते हैं यहाँ पे. ये जो है मसल्स के अगेंस्ट, हाँ मसल्स के मतलब in people with myasthenia gravis the body produces antibodies that block muscle cells from receiving messages from the nerve cells. यानी एंटीबॉडीज जो है न्यूरो ट्रांसमीटर्स के अगेंस्ट डिजाइन कर देता है हमारा बॉडी. न्यूरो ट्रांसमीटर्स लाइक एसिटाइल कोलिन के अगेंस्ट माइस्थेनिया ग्रेविस में एंटीबॉडी का प्रोडक्शन हो सकता है. तो दिस इज अ वेरी डेंजरस डिजीज. तुम लोग सोचो कि न्यूरो ट्रांसमीटर्स लाइक एसिटाइल कोलिन के अगेंस्ट एंटीबॉडी डिजाइन हो गया और वहाँ से मसल सेल्स जो नर्व सेल्स से मैसेज रिसीव करने वाला था वो सारा बंद हो. देन देयर कम्स पर्निशियस एनीमिया. पर्निशियस एनीमिया में ऑटो इम्यून कंडीशन इन द बॉडी वेयर द immune system attacks the intrinsic factor protein or the cells lining in the stomach that make it. So ये जो है ये विटामिन B12 के अगेंस्ट एंटीबॉडीज को डिजाइन कर सकता है और वहाँ से पर्निशियस एनीमिया हो सकता है. रिमोटेड आर्थराइटिस, kidneys, brain and other organs like the organs and also systems in which the DNA molecule is generally involved. Okay, so these are basics of the autoimmune diseases that take place in our body. So these are the 18 common autoimmune diseases like multiple sclerosis, Graves' disease, Hashimoto's disease, lupus, that is the systemic lupus erythematosus, psoriasis, Addison's disease. There are many types which are there, but I have just mentioned the important ones down here. There is the inflammatory bowel disorder, which is also an autoimmune disease, rheumatoid arthritis, okay, Addison's disease, celiac disease. So there are several diseases that are involved in the autoimmune section. So when we talk about leukocytes, we have to also mention these because they are important blood immune cells. So when we talk about leukocytes, we talk about the white blood cells. Now in our blood there are three types of cells which are generally we talk about, that is the red blood cells, the white blood cells and the platelets. Now keeping the red blood cells and the platelets aside, the most important cells of our of our blood in the form of in the form of the immune impact of our body is the leukocytes or the white blood cells. Now these leukocytes can be divided into granular leukocytes and agranular leukocytes. Granular leukocytes are the ones which comprise of different types of granulous materials. That means granules are present inside the leukocyte. They are the granular leukocytes, and where no granules are present, they are known as the agranular leukocytes. Granular leukocytes have certain examples, that is the eosinophil, basophil, neutrophil. But agranular leukocytes are examples in the form of lymphocyte and monocyte. Lymphocyte may be the T lymphocyte, B lymphocyte, natural killer lymphocyte or the natural killer cell, and monocyte macrophages, Kupffer cells, and there are several different types of classification that are available. Eosinophil, basophil and neutrophil are the granular leukocyte, and they comprise of even mast cells also. They comprise of huge number of granulated materials inside them. Okay. So this is basically the difference between all the types of the cells that are present in our body. Body immune system, that is the macrophage, neutrophil, eosinophil, basophil, mast cell, lymphocytes, monocytes and natural killer cell. So macrophage basically in adults has a very variable structure. This is basically, let's look into the function, though other parts are not important. The ones which is important is neutrophil has been found in the highest concentration in our body. Now macrophage functions in phagocytosis, antigen presentation to the T cells. Now neutrophil, I have already told that in case if you look into a blood report, you will find out that the most number of cells that are found in our body is the neutrophils, 45 to 75%. It is multi-lobe structure. See, many lobes are present, and counting the number of lobes inside a neutrophil has also been counted by a term that is known as the Arnett count. Okay, phagocytosis, degranulation, that is the discharge of the contents of a cell. Then there comes eosinophil. Eosinophil has 1 to 6% of the total percentage in our body. It is generally bilobe, so it's a headphone-like structure. There is a basophil, that is known as the bi or trilobe, release histamine, enzymes, and cytokines. Then there is the mast cell, release histamine, enzymes, and cytokines, is involved in the allergic responses. Lymphocytes is also present in 20 to 40%. So this is the second highest percentage of abundance that is present in case of the human immune system. Now there might be the cytotoxic T cells, the T helper cells that are present. Monocyte is generally 2 to 6%. It is generally kidney shaped structure. Differentiate into macrophages and dendritic cells to elicit immune responses, and they might be localized in different organs or they might be free rotating. Natural killer cells, now these are very much important in rejection of tumors. So they fight directly against the cancer cells. Okay, so these are the generalized cells of our immune system. So let's just look into the questions at the end of the session today. So here is the given that match the immunoglobulin with their function. Here is given IgE. So you know that IgE is involved in the immune allergic responses. So IgE, it mediates the allergic reaction. Then there comes the IgG. So the options given is in the forms of the protection of the fetus, first antibody to be produced in response to infection, provides localized protection of mucosal surfaces, mediates allergic reaction. We have already eliminated this option with the form of IgE, directly lyses the cell. So you know that the IgE antibody are produced generally at the beginning. That means it has ability to protect the fetus. That means they can cross the placental barrier and enter into the extra-fetal area, okay, inside the placenta. There is the fetus and the extra-fetus area inside the placenta, so they give a protection directly to the fetus. So they protect the fetus. Then there is the IgM antibody. We know that the IgM antibody are the first type of antibody that are produced at the beginning of any kind of immune response. So first antibody to be produced is produced in response to infection. Okay, so let's just give a match into all these things. So IgE, I have said S, that means 1S. 1S is present in case of only one option. Is there any other option? No. Let's match the other ones. 2P, okay, 2P, that is also good. IgG 3, that would be generally would be Q, 3Q. Okay, that's good. Oh, we have missed out with IgA. I'm very much sorry about that. IgA, now IgA generally, you know, I have already mentioned that the mucosal surfaces are generally protected. You can go back to the slides and you can see that the mucosal surfaces are generally protected using the IgA antibodies. There was a functional area where I have already mentioned this. So it would be the provides localized protection of the mucosal surfaces. So 4 would be R. So that's my yes, 4 is R. So correct option would be option number A. Okay, and direct lysis of the target cell. This generally occurs by the perforin and granzyme enzymes that are secreted by the CTLs or the cytotoxic T lymphocytes. If there would have been any option involving CTL, you should have correctly selected this option as T. Okay, so option number A is correct. Next is match the autoimmune diseases in column 1 to the self antigens in column 2. So rheumatoid arthritis is an autoimmune disorder. Now this involves generally the connective tissues, right? So rheumatoid arthritis जो हमारे connective tissues है, bones का है, उसका disorder होता है. Systemic lupus erythematosus. I mentioned the systemic lupus erythematosus involve the changes in the DNA. So option number 3. Myasthenia gravis. Myasthenia gravis के यहाँ पे जो है हमने बोला था कि इम्यून सेल्स ऐसे होते हैं या फिर एंटीबॉडीज ऐसे होते हैं जो न्यूरोट्रांसमीटर्स या फिर न्यूरोट्रांसमीटर के जो रिसेप्टर्स हैं उनको टारगेट करता है. तो न्यूरोट्रांसमीटर्स या फिर न्यूरोट्रांसमीटर के रिसेप्टर यानी कि लाइगैंड लेवल या फिर रिसेप्टर लेवल में जो टारगेट होता है उससे ही ये माइस्थेनिया ग्रेविस के नाम के एक डिजीज जनरली टेक प्लेस करता है. और उसके बाद जो है एसिटाइल कोलिन रिसेप्टर्स होता है, तो वो जो है मायोसिन ग्रेविस के रिलेटेड है। मल्टीपल स्क्लेरोसिस जो है वो माइलिन शीथ के अगेंस्ट एंटीबॉडी या फिर नॉट एंटीबॉडी, इम्यून सेल्स जो है माइलिन शीथ को टारगेट करके वहाँ पे इन्फ्लामेशन कॉज करते हैं, जहाँ से जो नर्व सिग्नल्स है वो ट्रांसमिट ठीक ठाक से नहीं हो पाता। सो लेट्स लुक इनटू द ऑप्शंस ओवर हियर. So you can find it over here that P would be 2. Okay, P2 only one option again. So you can look into one option and just correct it up. Systemic Q3. Okay, Q3, yes. Then R would be 4. Okay, R4 and A should be 1. Okay, so option number B is correct. So you can look over here. These multiple choice questions in case of match the following is very easy to attempt. Okay. So which one of the following leukocytes is phagocytic and has clear cytoplasm? So we know that the phagocytic cells are generally the macrophages. We have the monocytes. We have the B cells. Generally, they are phagocytic in nature and clear cytoplasm. So clear cytoplasm is present in case of the lymphocytes. Clear cytoplasm is also present in case of the monocytes. But T lymphocyte doesn't have phagocytic activity. They generally have phagocytic receiving activity. That is, they can receive information via the MHC generated fragments via the T cell receptor. ओके, TH lymphocyte won't be an option. बेसोफिल एसिनोफिल के पास कोई क्लियर साइटोप्लाज्म नहीं होता है, वहाँ पे ग्रैन्युलेटेड साइटोप्लाज्म होता है, तो ऑप्शन करेक्ट होगी मोनोसाइट्स. ओके, मोनोसाइट के पास फैगोसाइटिक एबिलिटी होता है, मोनोसाइट के पास क्लियर साइटोप्लाज्म भी है. सो ऑप्शन नंबर बी वुड बी द करेक्ट ऑप्शन. In the human body, which of the following is involved in the processing of a foreign antigen? So foreign antigen के processing में कौन सा वाला cells generally involved होता है? B cells, macrophages, red blood cells, platelets. So फॉरेन एंटीजेंस को प्रोसेस करने में जो ज़्यादा